So now I'm going to discuss with you about the organogenesis and somatic embryogenesis. In the previous video, I have just mentioned about the micropropagation process. Although organogenesis and your somatic embryogenesis are also can be considered as a technique related with the micropropagation, but the main source or the type of how they do culture is little bit of different. Okay, so normally organogenesis is the development of your adventitious organs, or you can say the primordia, from the undifferentiated cell mass in the tissue culture by the process of differentiation. Now, this organogenesis, as it is saying in this book, that it is a process of morphogenesis, structurally change that involves the formation of plant organs like root formation, flower buds from the explant or cultured tissues. Organogenesis are of two types: direct organogenesis and indirect organogenesis. Direct organogenesis simply means that directly from the explant there will be some morphological changes, there will be some morphogenesis without undergoing any callus formation. They will directly form the adventitious organs from the explant. So it is saying that tissues from the leaves, stems, roots, and inflorescence part of the plant can directly be cultured to produce the plant organs. In direct organogenesis, the tissues undergo morphogenesis without going through callus or suspension cell culture stage. So there will be no callus formation. Directly, the explant will be used, and this explant will be it will morphologically it will change to form the adventitious organs of the plant, the desired adventitious organ that we want, like shoot from that explant or root from that explant, okay, or the other parts as well. So induction of adventitious shoot formation directly on roots, leaves, and various other organs of intact plants is a widely used method for plant propagation. This approach is particularly useful for herbaceous plant. So directly here, without undergoing any callus stage formation, the the adventitious organs are developed from the explant. Like for example, if I want to develop adventitious shoot formation from the root explant, suppose I have taken root as an explant, and from that root I have inoculated into a particular media. That root undergoes some morphological changes, morphogenesis, to form adventitious shoot formation. That is your direct organogenesis. Now, for appropriate organogenesis in culture system, we have to add externally. Like that's why it is written that exogenous means externally from outside. We have to add the growth regulators in certain concentration, like auxin and cytokinin is required. The concentration of the growth promoting substance depends on age and nature of the plant, besides the growth condition. So you can see the plant, its leaves, the leaves are cut into explants. And if you see the number A column, not the B or C, in the A column you can see directly from the leaf the adventitious shoot formation has been done. No callus formation takes place. The B and C figure, as you can see, callus formation takes place. They have undergone suspension cell culture, so they can be considered as indirect organogenesis. So the direct organogenesis is your this part, okay, this part where you can see that directly shoot formation is occurring from the leaf substance, okay. Then after shoot formation, rooting, they are placed into certain subsequent subculture is being done for the formation of root in that culture, in that plant, and subsequently we are getting that whole plant. So that is the direct organogenesis. Now this type of concept of direct organogenesis has been get from the idea of certain plants like begonia plants, which then bryophyllum plant, which develop its plantlets from the edges of their leaf blades, right? So this type of concept it comes. Let us see now about the indirect organogenesis. As I have stated that in indirect organogenesis, they have to undergo callus formation, or they can go for suspension culture after callus formation. The callus is broken into pieces, the friable callus, and they are further agitated in a liquid media for development of the callus. And from that callus, we will develop the adventitious organs. So indirectly we are taking some more steps, that is your indirect organogenesis. So when the organogenesis occur through callus or suspension cell culture formation, it is regarded as indirect organogenesis. So here callus formation will be occurring, and from that callus we will grow the adventitious shoot root, much more or less like your micropropagation technique. That's why the organogenesis is also a part of micropropagation. So callus growth can be established from various explants, like from the leaves, from the roots, from the cotyledons, stems, flower petals, etc. Now, the explants for good organogenesis should be mitotically active in mature tissues. This has to be taken into consideration. So, this part that I am going to say, this part has to be it is very, very important that the explant good for organogenesis should be mitotically active in mature tissues, right? So it should be mitotically active in mature tissues so that they can proliferate to grow into certain adventitious organs of the plant. Now, in general, the bigger the explant, the better the chance for obtaining viable scalars or cell suspension culture. So, if the explant size is much more, then greater the chance for survival for obtaining viable callus. But at the same time, we can make that explant genetically variable, because non-meristematic parts of the explant can leads to formation of indoor reduplication concept, and that leads to the formation of chromosomes with four chromatids, eight chromatids, polytene chromosomes. Polytene like conditions will occur. So chromosomal variation can occur if I take a big explant. At the same time, there is a chance of pathogens entry. That's why we should take only the meristematic parts. Okay, like shoot, tip, leaf, petiole. Okay, they are sufficient for indirect organogenesis. Now, for indirect organogenesis, cultures may be grown in liquid medium or in solid medium. And many tissue culture media like your Murasijan's cook media, then B5 media, then White's media can be used in organogenesis. The concentration of growth regulators in the medium is very critical for organogenesis. If I vary the concentration of this growth media, in vitro organogenesis can be regulated, it can be manipulated. So the same thing has been written again and again, like I have in the first class I have mentioned with you about this hormonal concentration of this phytohormone concentration, that low auxin low cytokinin will leads to callus formation, low auxin high cytokinin will leads to shoot organogenesis from the callus, high auxin and low cytokinin will leads to induce the root formation. So this is the indirect organogenesis. As you can see, that same procedure, but they have to go through callus stage. Now if the callus is friable, if it can be easily breakable down, they can be broken into pieces, and they they are subjected to for suspension culture, where the small friable callus are grown on liquid culture medium, okay, without using any agar. And by properly agitation means mixing of the culture medium with the help of rotary shaker, we are constantly growing the callus tissues into big lumps of callus, which can be further subcultured to generate shoot formation, as you can see, by placing them into such medium where low concentration of auxin and high concentration of cytokinin is present. Then we place them in rooting medium, the same as micropropagation. Don't be worried about this part. It is all almost same. The main objective is how they are being prepared. Okay, and finally to grow into a plant. Okay, so this was about the organogenesis. Okay, okay, so now we are going to discuss about somatic embryogenesis. Now, this look like strange thing that we can generate embryos from the somatic cells, that is the normal cells of the explant, from the tissues, from organs, that is known as somatic embryogenesis. So it's a strange thing that how we can develop embryos from the somatic cells. Yes, it is possible by various combinations of growth hormones, growth regulators that we normally use. Even some of the composition that is mentioned, that is normally we using in the culture media has to be a little bit changed in their quantity. Okay, to get that beneficial result. So it is saying that the process of regeneration of embryos from the somatic cells, from the tissues or organs, is regarded as somatic embryogenesis. Somatic embryogenesis is also a type of asexual reproduction. That's why we have categorized them and they have mentioned in this micropropagation chapter. Somatic embryogenesis may result in non-zygotic embryos or somatic embryos. So it is not just like sexual propagation where there is a contribution of the genetic material of the male plant via the pollen grain and the female plant via the ovum, but it is directly non-zygotic embryos are formed, directly formed from the somatic cells of the explant. Now this is taken from different source. I will mention with you to you that where I have taken the source from, where in the WhatsApp group. So let us discuss more about this somatic embryogenesis and how they are produced. They are also of two types: direct somatic embryogenesis and indirect somatic embryogenesis. We will mention them just a bit later. So what it is saying here, let us see. It is saying that a somatic embryo is an embryo derived from a somatic cell. Okay, other than a zygote. So, other than a zygote, we are producing embryo from somatic cell and obtained usually on culture of the somatic cells in vitro. This process is called somatic embryogenesis. Okay. Now, in zygotic embryos, we know that there is a contribution of the genome of your male and female. But here in somatic embryos, they are fully your embryos developed from the somatic cells. No sexual reproduction. Okay. Now, development pattern of somatic embryos, or SE. It is saying that somatic embryos generally originate from single cells, which divide to form a group of meristematic cells. So this is the line. If you see, if you see that somatic, okay, I have to. Hmm, it's quite thicker. Okay, now it's okay. So somatic embryos generally originate from single cells which divide to form a group of meristematic cells, right? And usually this multicellular group of meristematic cells become isolated by breaking their cytoplasmic connections with the other cells. So you can see that it is saying that usually, again, okay. So it is saying that usually this multicellular group of meristematic cells become isolated by breaking their cytoplasmic connection with the other cell. So it means to say, just like this. Okay, I'm drawing a diagram. There is a diagram after this also. Suppose it is a group of cells. These are group of cells, and just in that there is a meristematic cell. Suppose there is a meristematic cell, okay, written as short form as MC. Now this meristematic cell, what they are saying, this multicellular group of meristematic cells. So this group of one meristematic cell I have said here, I have mentioned, I have drawn, but it is saying this multicellular group of meristematic cell become isolated by breaking their cytoplasmic connections with other cells around it, and subsequently by cutinization of the outer wall of this differentiating cell mass. So they form a cutinization. Cutinization is normally, as mentioned here, handwritten, it is saying, here you see here in this part, cutinization is a process. Okay, cutinization is a process in which the outermost plant cells become thickened and covered with cutin, making them waterproof. So cutinization occurs, the cytoplasmic connections with the other cells, they become isolated by breaking the cytoplasmic connection with the other cells around it, and subsequently cutinization occur, where the outermost plant cell become thickened and covered with cutin, making them waterproof, right, making them waterproof. Now the cells of this meristematic mass, they will continue to divide and give rise to different type of shapes like globular shape, heart shape, torpedo shape, and cotyledonary stage of somatic embryos. So in the next page I'm trying to show you the diagram, you can able to understand this part. So in the first figure, as you can see, in the first figure you can see in this figure that initiation of somatic embryo development occurs, and this is the meristematic cell. Okay, this is the meristematic cell. In this way there are many meristematic cells in that part, and this multicellular group of meristematic cells, they break their cytoplasmic connection. As you can see here, they are breaking their cytoplasmic connection here and there, and with the other cells, and they subsequently form a cutinization. This part is cuticle, which is a protective layer, you can see, that formed around the mass of cells. And this group of cells which forms a cuticle inside, they give rise to different shapes. As you can see, the first shape is your globular shape. This is a globular shape. Okay, then it becomes a different shape, that cell mass of mesotic cell become heart shape, and then subsequently into torpedo shape. Okay, this is a torpedo shape, as you can see, and finally you can see a cotyledonary stage. So simple thing, the meristematic cell mass, they break their cytoplasmic connections with the other cells, and they form cutinization, where a cuticle formation occurs made up of cutin, and the cytoplasmic connections are broken down to form a clump of cells, group of cells, and subsequently these cells of meristematic mass continues to divide and give rise to different shapes. As you can see, globular shape of meristematic cells, then your heart shape, then torpedo shape, and then cotyledonary shape, which is the cotyledonary stage of somatic embryogenesis. So in general, the essential features of somatic embryogenesis development or somatic embryo development, especially after the globular stage, are comparable to those of zygotic embryos. So simple thing, different stages can be observed. Although this is not the real diagram, the real image, I will give you, I will provide to you in the WhatsApp group, that how these meristematic cells, they break their cytoplasmic connection with the other cells and form cutinization, forms a thick cuticle around it, And inside the cuticle, this group of cells, they break the they break their cytoplasmic connections and form different type of shapes like globular shape, heart shape, torpedo shape, and cotyledonary shape, which is the main final shape of embryo, which can be compared with the zygotic embryos as well. Now, the somatic embryos are bipolar in structure. They are bipolar in structure. That is, they have a radical and a plumule. So, somatic embryos have a radical and a plumule. The radical of the not only somatic embryo, in every embryo they have a radical and a plumule. The radical will give rise to root formation. It will makes up the root system of the plant, while the plumule part that emerges after the radical, it makes up the shoot. Generally, the radical formation occurs first, and then the plumule development occurs. But in vitro culture, we are eager to grow the shoot part first and then the root part. So that's different issue. But here you can see the radical and the plumule part, which give rise to the root system and shoot system respectively. The radicular end is always oriented towards the callus or the cell mass, and while the plumular end always sticks out from the cell mass. So it is given in the figure 8.5. Uh, in the figure 8.5, if you see the figure, it is saying that the radicular end which will makes the root formation, they are oriented towards the center of the cell mass. It has not been shown that where is the radical, where is the plumule, but this radical part is actually oriented towards the center of the callus cell mass, while the plumular end, which will give rise to make the shoot system of the plant, it always takes out from the cell mass. Okay, so this is the plumular end and this is the radicular end that we can see here. Okay, then next it is saying that in contrast, shoot bud is monopolar and in it does not have radicular end. In many somatic embryos, radical is suppressed so that they often do not produce roots. In such cases, roots has to be regenerated from the shoots by produced by germinating somatic embryos. Now, for overcoming this problem, we can you can use presence of abscisic acid, or you can use the abscisic acid, also in short we known as ABA, or suitable concentration of mannitol solution. So it is written here in this part that somatic embryos that we develop from the somatic cells often show some abnormal developmental features like three or more cotyledons, okay, bell shaped cotyledon, large embryo size. These problems can be solved by using quite amount of abscisic acid or suitable concentration of mannitol. Now, in some species, normal looking somatic embryos are formed, but they fail to germinate. That means they become sterile. At least some somatic embryos do not germinate in most of the species. So, we have to grow cultivating more and more somatic embryos so at least some of them can generate into a new plant. Okay, so in this way, somatic embryo formation occurs. Okay, let us see the other things that has been mentioned here. It is saying that somatic embryogenesis, this part, yeah, this part. So it is saying that somatic embryogenesis is influenced by several factors like growth regulators, like which has been said by GR, exposure to stress. If I place the plant in a if I culture the plant in stress conditions such as nutrient depletion, means less amount of nutrients available in the culture medium leads to a stress condition, can also leads to the formation of somatic embryos as shown in the research papers. Then arabinogalactan proteins, which are also called as AGP, we have to see them, what it does, what it plays a role in somatic embryogenesis. Then nitrogen source, explant genotype, and other factors are there. Some of the factors I will discuss, like for example, growth regulators. So growth regulators in most species and auxin, generally we use 2,4-dichlorophenoxyacetic acid at zero point 0.5 to 5 milligram per liter of your medium is essential for somatic embryogenesis. So this has to be noted if you want to do practical based on somatic embryogenesis, that if I add auxin to the medium, to the culture medium, generally 2 to 2,4-D, that is 2,4-dichlorophenoxyacetic acid, at zero point 0.5 up to 5 mg per liter in the culture medium, per liter of culture medium, if it is given, then it is essential for somatic embryogenesis. Auxin actually causes de-differentiation of a proportion of cells of the plant. Now, what is de-differentiation? Auxin promoting de-differentiation of the explant cells. De-differentiated cell is the process where the differentiated cell can regain its capacity for cell division under certain conditions, and the phenomenon is called de-differentiation. I have already mentioned with you in the first class of our plant tissue culture lecture that auxin, I have not said that auxin promotes de-differentiation, but I have said the definition of de-differentiation where the cell regain its ability to differentiate again under certain condition by mitosis process. Okay, so the auxin is promoting de-differentiation of a portion of cells. Those cells which not have lost their potency of doing cell division, they will start to divide, which begins to divide. In carrot, these small compact cells divide asymmetrically, and the daughter cells stick together to produce a cell mass called embryogenic clumps. So it is saying that in case of carrot, these small compact cells, they divide asymmetrically, not in a symmetrical manner, not in a regular pattern. That's why the daughter cells, as they divide asymmetrically, they stick together and produce a cell mass called as embryogenic clump, or also known as pro-embryogenic mass. In presence of auxin, these embryogenic callus, these embryogenic clumps grow and break apart into small cell mass, just like callus, which again produce embryogenic clumps. But when auxin is either removed or reduced its concentration, that is zero point zero 0.01 to only zero point 0.1 milligram per liter of solution, we are adding auxin, that like 2,4-D, the cell density, and if the cell density is lowered, each embryogenic clumps, which is known as EC, will give rise to few several somatic embryos. So simple thing, it is saying in carrot plant, they we use the 2,4-D as auxin source at a concentration of zero point 0.5 up to 5 milligram per liter of your culture media. It is showing that when applied, these carrot plants, they divide asymmetrically, and the daughter cells stick together and produce clumps called embryogenic clumps. No, in short, we can write as EC. In the presence of auxin, these embryogenic clumps will grow and break up into small cell mass, and again they will form embryogenic clumps. To overcome this problem, we can reduce the concentration of auxin, or we don't apply auxin to the culture medium, and if the cell density is lowered, then these embryogenic clumps will give rise to somatic embryos. Now, somatic embryos is believed to develop from single superficial cell of the embryogenic clumps. So that's why the ability to regenerate, that is of somatic embryos, the totipotency characteristics is acquired by the cell during de-differentiation in response to high auxin content. Okay, that's why we are adding auxin, so that the cells get their ability and carry out their de-differentiation process, where they can have their ability to differentiate into by mitosis in this condition. Auxin, it has also been mentioned that they also promote hypermethylation of DNA. Now, this hypermethylation of DNA, you have learned in your molecular biology class that uh, certain changes in the structure of DNA by addition of some functional groups can also regulate the gene expression. So hypermethylation normally plays an important role in tissue specific transcription process. Transcription, as you know, the process of formation of RNA from DNA used as a template. Now, by methylating the particular regions of the DNA, we can able to regulate the transcription process in specific tissues. So auxins also play an important role in this hypermethylation process. Then is saying that in many species like carrot, coffee, alfalfa, somatic embryogenesis is a two-step process. Okay, like so this type of plants, carrot, coffee, alfalfa, somatic embryogenesis is a very simple process in this case. Somatic embryo induction occurs on high oxygen up to 40 to 60 milligram per liter of 2,4-dichlorophenoxyacetic acid is used. So in case of these plants, we can use the 2,4-D as previously mentioned, but it is saying 40 to 60 milligram per liter, quite more compared to the previous one where we use only zero point 0.5 to 5 milligram per liter of oxygen. Somatic embryo development takes place on low oxygen or GR free medium. That means growth regulator free medium can also be used. It has been shown in the research that without using oxygen also some of the plants like coffee, alfalfa can grow into somatic embryos. Okay, so many things are written here. I am not saying all of them. And okay, so this was about the hormones. Next is exposure to stress. I think I have not. Okay, I have cut off this portion. Some of the portion has been written and above. So exposure to stress means some evidence have said by practical experiments that physical factors in the culture environment also plays an important role for induction and development of somatic embryos and their conversion. Like it is saying that in case of carrot, if the period of starvation occurs, starvation, as you know, depletion of nutrients in the culture medium leads to starvation. It shows that particular explant taken from the carrot, embryogenic cultures can be increased. Culture of embryogenic calli means callus, on half strength ms medium. Half strength ms medium means all the contents that are used in the ms culture medium, the composition of the contents, the composition of the constituents are being used half. That means if we use 8 gram of sugar as a carbon source per liter of solution of ms medium, here we will use 4 mg. So half strength ms medium means this. So if we culture the somat embryogenic callus on half strength ms medium, then also or medium lacking sucrose. Okay, if the medium is lacking sucrose also, or if the humidity is reduced, then there is a chance of increased somatic embryo formation for a factor of 3.4 to 4.5. So it has been shown by evidence that if we reduce the strength of the constituents that are being used in the molasses cooked medium and grow the callus, grow the explants, then there is a formation of rapid formation of somatic embryos. Or if we don't use any sucrose, then also um, there is a chance of formation of a somatic embryo formation. But starvation of embryo, it is written now here, but starvation of embryogenic calli for 5 days by culturing them in 12 well plates without nutrient medium enhanced embryo production by 20 fold. So it is saying that if the embryogenic callus, that means from the callus we will develop the embryo, that embryogenic callus, if they are staying in starved condition, absence of nutrients for 5 days, and culturing them in 12 well plates but without any nutrient medium, it shows that they enhance the somatic embryo formation by 20 fold, 20 times they grow well. Somatic embryos are grown. It also improves the somatic embryo maturation and germination. Now, in some wheat, somatic embryos was promoted by 40 millimole of NaCl and KCl, but this effect was genotype dependent. That means in the wheat plant also, somatic embryogenesis is made possible by promoting some culture, by promoting some chemicals, like we have added 40 millimolar of NaCl and KCl to the culture medium. But it is showing that it fully depends on the genotype dependent. It fully depends on the gene content, the genotype of the wheat, what family or what species of wheat we are using. The next factor that widely affect the somatic embryo formation is arabinogalactan proteins. What is arabinogalactan proteins? As the name suggests, galactan, so it is must be a glycoprotein, means carbohydrate with protein. Some glycoproteins produced by totipotent cell mass are secreted into the medium, and these proteins are added to the culture medium. They speed up the process of acquisition of totipotency. Acquisition means acquiring the totipotency part. That means they will form a whole plant. So there are some glycoproteins that are produced by the totipotent cell. Now each of the cell of the plant are totipotent, that we know. So they release the glycoproteins, and these glycoproteins, when if these proteins are isolated and give separately to the culture medium, they speed up the process of somatic embryogenesis. A class of protein called arabinogalactan protein, consisting of 90% carbohydrate with protein backbone, seems to be involved in somatic embryo formation. This arabinogalactan protein, also known as AGP, they are heterogeneous group of proteoglycans. Means they are not uniformly constitute part. They are of different groups, and they are found in plasma membrane. It has been shown that when extracellular, um, from here you can see, I am marking it, okay, from here. So it is saying that when extracellular, these arabinogalactan protein are secreted to the culture medium by embryogenic suspension culture of carrot and some other plant species, where insolated, means to place in sunlight, and added to less embryogenic suspension cultures, the embryogenic potential of the latter was markedly increased. So arabinogalactan protein seems to play an important role in early stages of somatic embryogenesis. So we can add externally these arabinogalactan proteins, which are generally secreted from the explants, from the totipotent cells. But if we add externally to the culture medium and grow the our desired tissues, desired explants, then we can get a rapid somatic embryo formation from such explants. Then it is showing the nitrogen source. Nitrogen source normally it has marked effect on somatic immunogenesis. It has been says variously experiments has been done, like for example, presence of low level of NH4 ammonia in carrot plant, 10 millimole per liter in combination with NO3 minus is required for somatic embryogenesis regeneration. So nitrogen source is also a very good source which promotes the somatic embryo formation as given in this paragraph. It is saying that the form of nitrogen has marked effect on somatic embryogenesis. In carrot, in carrot the ammonia has promotive effect on somatic embryo regeneration. In fact, induction of somatic embryos in carrots occur only when about 5 millimole per liter of cell fresh weight ammonia presence is present in the cells. So ammonia in combination with the NO3 source is the well that can be used for generation of somatic embryos more rapidly. Let us see the direct somatic embryogenesis and the indirect somatic embryogenesis. Okay, so the direct somatic embryogenesis. So as the name suggests, we will not develop any callus formation here. So when the somatic embryo develop directly from the excised plant, the cut off plant, which is explant, without undergoing callus formation, that is called as direct somatic embryogenesis, and is possible due to the presence of pre-embryogenic determined cells. So there are some cells called pre-embryogenic determined cells, which are actually the mesosomatic cells, which has the capacity to being divided to form that has a capability to undergo cutinization and undergo several stages, several morphological changes to form the somatic embryos. So this is the direct somatic embryogenesis process. You can see here. So this is the explant, and after the explant, you can see the petiole has been taken. Petiole has been taken. Petiole is the stalk. It is the stalk that attaches with the leaf to the plant system, and then we take away the explants, take the explants from that part of the plant, where we can undergo the direct somatic embryogenesis as well as indirect somatic embryogenesis process. Okay, so these are plantlet with roots, same thing, and that's the plant. Now the question arises that after the somatic embryo formation, what we will do? What we will use the embryos? How we can use these embryos for plant generation? Well, simply we can go in a many way, like we can produce artificial seeds, right? We can produce artificial seeds from the somatic embryos that can be made by encapsulation of the somatic embryos. Embryos are coated with sodium alginate and the nutrient solution. They are dipped in calcium chloride solution. So simple thing, we use a beaker here. Okay, and then with the help of micropipette, with the help of micropipette, we will add drop by drop. We will add drop by drop to the solution. Now this pipette consists of sodium alginate and the nutrient solution. I am writing in short form: sodium alginate and the nutrient solution with the embryo. And in this beaker, it contains calcium chloride solution. Now, when we are dipping this by the help of the pipette, when we are dipping this mixture, the sodium alginate with nutrient medium with the embryo in the calcium chloride solution, there is a polymer formation, a rapid cross-linking of sodium alginate occurs that produce small gel beads, and each bead consisting of encapsulated embryo, just like this. Just like this it occurs. Okay. Now, these artificial seeds can remain for a long time because it is a polymer substance. It can oxygen can enter easily and comes out. At the same time, it is getting the nutrients as we have used the mixture of sodium alginate with the with the nutrient medium along with the embryo. They were mixed properly, and then they are being dipped into the calcium chloride solution where they form a cross-linked polymer to form these artificial seeds. These artificial seeds are further dried then and then used as normal seeds for a long period of time. So in this way, we can commercialize uh, in this somatic embryo formation, and we can get a true to type plantlet from taken from the mother plant. So this was all for today's lecture. I hope you have understood this part. If you have any doubts regarding this lecture, you can comment me in WhatsApp group. Okay, and I'm trying to send you the photos of your these the stages of somatic embryo formation. Okay,